Right. I guess we should carry on, should we? Final video of Unit 7. Then it's the last unit, the DMT unit. Yes, so as I said, I want to just talk a little bit about some of the neuroimaging work, which is, it's been absolutely transformative, transcendentally transformative in understanding psychedelics. I kind of think of myself primarily as a kind of molecular scientist. I'm really fascinated at what goes on at the subcellular level and the level of individual neurons and stuff. And of course that's really important work, but it's also important to kind of understand psychedelics at all these different levels of organization, as well as on a subjective level as well. It's important to understand people's experiences. The phenomenological aspects of psychedelics are also important, and what science is doing now after really several decades long hiatus because of ridiculous drug laws. Don't get me started on that. But anyway, so it's important that you look at the effects of psychedelic drugs at many levels of organization. And as I said, I would be remiss if I didn't talk about some of the kind of pioneering functional neuroimaging work that's been carried out, I dare say mainly at Imperial College London by the Psychedelic Research Group headed up by Professor David Nutt and Dr. Robin Carhart Harris, who really are, really a huge team of colleagues and collaborators really around the world really now, who are observing the effects of psychedelics and able to analyze the effects of psychedelics in living human brains with psilocybin initially, then LSD, and we'll look at that study particularly, and then more recently DMT as well, which we'll look at in the next unit. Okay, so this is the study that I'm going to look at just a little bit. So this is a study that came out a few years ago now, that actually looked at, this was from the Imperial College team, as I said, the leading team really, so Dave Nutt and Robin, who head up this team. And this looked at the neural correlates of the LSD experience revealed by multimodal neuroimaging. So what does that mean? So what these studies normally do is use something called functional neuroimaging. So functional neuroimaging is a type of magnetic resonance imaging, or MRI. So you put somebody into one of these huge MRI machines, and you're not really detecting brain activity as such, but you are detecting changes in blood flow and oxygenation levels of the blood as the brain is working. So basically, as different parts of the brain are working, are activated, they tend to draw blood to them, oxygenated blood, and using what's called a blood oxygen level dependent signal, you can actually detect these very, very tiny changes in blood oxygenation and blood flow in these areas of the brain. So that's really what these studies are detecting, is the changing in this so-called BOLD signal, blood oxygen level dependent signal. And as well as, this is when you see these kind of brain images of different parts of the brain kind of lighting up. Really, they're not really lighting up, of course. This is actually showing this particular signal that's detected by the MRI machine. And what's beautiful about this technology is that you can not only see which parts of the brain are active, but you can using statistical techniques, you can see how different parts of the brain are connected. So when this part of the brain is active, let's say, this one also seems to light up at the same time, or when this one is active, this one always shuts down, or when this part is active. A little bit later, this one is always active, followed by this one. So you can get an idea as to how brain activity is not just kind of observing brain activity, but also observing the organization of brain activity, which allows you to kind of infer and conclude a lot about the way these brain networks, which we discussed in the last video, are organized. So before we look at some of the data, we should think a little bit about, we should kind of just make sure we're clear on what we already know about psychedelics, or what we've kind of concluded from our analysis of the way psychedelics affect the brain, i.e. that psychedelics tend to cause networks of columns to kind of break down and for information to flow out of these networks, basically generating a more kind of disordered, one would say, type of brain activity. And this happens both at the levels of networks of columns, but also, as we've seen, also networks of brain areas, such as the DMN, the default mode network, or the task positive network, for example. So let's actually have a look at some of the results from this study. So what we're seeing here is an image from that paper, that landmark paper on LSD imaging, and what this particular image is showing is what's called resting state functional connectivity, RSFC. Now, functional connectivity doesn't necessarily mean that brain areas are directly connected, by synaptic connections or whatever, but that they tend to light up at the same time during a particular task or not a task. So we looked at the default mode network, for example. These areas will be sort of functionally connected when you're kind of lying and closing your eyes and just reminiscing or letting your mind wander. You get sort of functional connectivity between these brain areas. So you have to do further experiments to determine whether these brain areas are actually directly influencing each other or not. But we're not going to get into that. And what this image is particularly showing is resting state functional connectivity, particularly with V1. So we know that V1, this area at the back of the brain, which receives sensory information from the environment via the retina and then the thalamus. And so this image is basically showing, which areas of the brain are connected to V1. And normally, of course, you would expect functional connectivity between V1 and V2, V3, V4, these other higher areas of the visual network that we've looked at. And indeed, you tend to see that. You can see that in some of these images. And there are other brain areas that are connected as well. But this top set of images shows the kind of the baseline condition. So someone who hasn't taken the drug, someone who's basically taken a placebo, a sugar pill. Whereas in this second line of images, we actually see the LSD condition. So again, it shows resting state functional connectivity with V1, but now you can see that V1 seems to be connected to a lot of different areas of the brain. And then in the bottom, you can actually see the difference between the two conditions, between the sort of no drug here and the LSD condition. So this is the difference between them. And clearly you can see there's an increase in the way that V1 at the back speaks to these other areas of the brain here. Yeah? So we talked about that, right? We said that when in the presence of a psychedelic drug, these networks start to break down and information starts flowing out of these well-established networks and brain areas that didn't used to speak to each other, or don't normally speak to each other, start to speak to each other. So there's a more of a disorganized, yeah, okay, that's fine. Disorganized got a negative connotation, but yeah, I think you get the idea. There's a more of a free flowing of information between brain areas. And so this is exactly what you observe in living human brains. And we were able to kind of derive that idea just from looking at, from first principles, basically, which is kind of cool, I think. Another thing that we discussed was the increase in the number of states, possible states, which kind of follows from the idea of this of information flowing out of cortical columns, and and more cortical columns speaking to each other, and I think I described it as like a democratization of brain states, where you get an increase in the number of possible states of the system, which of course increases the information when the brain selects a particular state. This as well has been observed and indeed quantified again by the same team at Imperial College London, and we can see that here. So this was a paper published again by an analysis of that. I think it was analysis of one of their earlier studies with psilocybin here. So in this study, what they actually looked at was what's called the entropy of brain activity, which is the level of disorder, if you like, in brain activity. And what they found, we won't go through all of these, we'll just look at this one, let's say, is that with when psilocybin is present, so this red line here, you see this higher level of disorder in the brain compared to the normal kind of waking state, which suggests that the brain is able to adopt a larger number of possible states and moves through these states kind of more randomly over time, which is exactly what we suggested would happen. And they were also able to actually quantify this somewhat and actually see which states, which kind of new states the brain was able to adopt. Now, they didn't look at states of kind of patterns of columns because MRI simply doesn't have the resolution to look at that. But they looked at patterns of brain areas, if you like. And what they found is that there was these kind of new states, new patterns of brain area network activity that were only seen in the presence of psilocybin. Again, exactly what we suggested would happen, and from first principles, kind of showing that psilocybin does indeed induce this state in which the brain is able to adopt a larger number of states. Great. Okay, so that was just a couple of kind of important studies. I mean, we could spend a whole course going through all of the psychedelic, the new psychedelic science, but we don't have the time. One thing I do want to talk about before we actually finish this unit, because this is going to be the last video, is something that sometimes confuses people a little bit. And we've said that psychedelics induce this slightly disorganized, more kind of free-flowing, dynamic state of consciousness. And people sometimes say, well, is that a good thing or a bad thing? Is it good or bad that the brain has entered this more dynamic and free-flowing state? And in some ways, it seems good. You have this wonderful experience. Your old ideas and old ways of thinking are kind of broken down. You enter this kind of highly kind of creative state. The world looks kind of magical and beautiful, and it's full of information, and you're overwhelmed by information flowing into your brain, and it's like, wow, I wish I could live like this forever. And some people might like that, but there is a cost to this. When I say there's a cost, I mean there is a cost during the experience. So the brain has to kind of balance order and disorder. The brain has to be able to organize its activity. That's the only way that you could have a stable and predictable model of reality that actually makes sense and that is functional. Your brain has to organize its activity to make sense of the world and for you to actually function in the world and to make predictions and navigate the world. At the same time, this model has to be very, very dynamic. You have to be able to account for the fact that the world is constantly changing and that there are ambiguities in the environment, and you may need to make quick decisions and then change your mind. You need to change your attention, for example, from looking outside to looking inwards and perhaps thinking through some kind of problem before making some decision on how to behave next. You need to solve problems. You need to change the way that you think about something in order to solve a problem, to get round something that's been some kind of task that you can't work out. Always used to work, and I for some reason I simply can't solve this problem. I can't do this. You need to change the way you're thinking. So you need this creativity and dynamic, and the malleability, if you like, of brain activity is absolutely crucial. But at the same time, there has to be some order. So it's a balance. Your brain is constantly sitting kind of the edge, really, of order and disorder, as all living systems are, really. And I must strike that balance. And what psychedelics do is they tip you slightly towards disorder. Not too much. Not so much that you completely lose any kind of sense of reality, although that can kind of happen with high doses of psychedelics. But the world is just nudged in the direction of disorder. And that opens up new ways of looking at the world. It opens up creativity. It makes you literally see the world anew, with entirely new ways of interacting with the world and navigating your environment, of simply being within the world. All of that is opened up by psychedelics. But what the cost of that is a loss of control over the organization of your brain activity, the organization of your thoughts, your ability to organize the contents of your subjective experience, your subjective world. And that's a wonderful thing to do, to kind of lose that control for a while. But then, you couldn't live like that, I think, permanently within that state, because you wouldn't really be able to function. So is this loss of control, this disorganized state, is it a bad thing? No, but it's not an optimal functional state. So it's something that we enter from time to time in order to kind of shake up the old model and develop new ways of thinking and interacting with and being within the world, for creativity or things like that. Okay, I think that just about covers it. So we have completed now this psychedelic neuroscience unit, where we actually brought together everything we've developed, all these ideas and concepts which we've developed over the last few units. So you should be very pleased with yourself if you've made it this far and actually gone through all of these units and understood everything. You are now really an expert on the effects of psychedelic drugs, and it's a wonderful thing when you finally, it's no longer as mysterious. They're still mysterious. They're still mysterious, but not as mysterious. And there are ways to kind of understand the way that psychedelics work. Then there's DMT. So in the next unit, we're gonna go into that strangest of strange drugs, that great reality channel switch, as Terence McKenna used to call it, dimethyltryptamine, and that will be the last unit. So get ready for DMT. I'll see you with the elves in the dome next. See you the next unit. kidding. However, if you do want to support the production of future courses, or you just want to leave a tip, then there are virtual tip boxes below. There's PayPal, Ko-fi, or Ko-fi, whatever it is, even a Bitcoin address if that is your thing. Minimally, please like and subscribe. It really helps me. And please follow me on Twitter and Instagram if you use them, Alien Insect. And I think that's about it. I hope you're enjoying the course. Thank you.